This video will cover subtopic 2.8, cellular respiration. Here's a list of understandings, applications, and skills that will be covered in this video. So to start it off, just a reminder that life takes work. The processes of life require energy. And where does that energy come from? It comes in the form of ATP. And how is ATP made? Well, ATP gets made through cellular respiration. So to start off, we'll take a look at ATP, that adenosine triphosphate. Having a look at the molecule itself, the part that contains nitrogen, this here is our adenosine, which is attached to a ribose sugar, and that ribose sugar is attached to 3 phosphate groups. That's where the triphosphate comes from. Now, there is a high amount of energy held in the bonds between the second phosphate and the third phosphate. So cleaving the bond between the second and third phosphate releases energy. And so for this reason, ATP is considered the energy currency of the cell. So what is cellular respiration? Well, cellular respiration is an almost universal process by which organisms will utilize the chemical energy in food to produce enough ATP to perform all the necessary actions required of living creatures. So it turns out that it is organic compounds that have to be broken down to release this energy, or that gets used to create ATP. Now, glucose is the primary biomolecule, or the primary molecule that is broken down by cellular respiration. There's a lot of energy contained within the bonds of glucose, and by breaking those bonds, that energy gets released and can be harnessed to use to generate ATP. So if you think about it, if ATP were quarters, glucose would be like a $10 bill. There's a lot of energy in glucose. Other organic molecules can be converted to glucose for energy harvesting, or other molecules can be used to generate ATP at different points in cellular respiration. So fatty acids can be used, as well as proteins can be used as energy sources. So those organic compounds ultimately, as I said, do have to be broken down to produce ATP. So for example, a molecule like glucose, in the presence of oxygen, will be broken down in a series of small steps to produce carbon dioxide and water. And as glucose is broken down in those small steps, you'll have ADP, which is adenosine diphosphate, a molecule that contains 2 phosphates instead of 3, that will react with a lone inorganic phosphate group that will react together to join or to create ATP, so in which that 3rd phosphate gets added on to the molecule, creating that energy-rich ATP. And so this process of cellular respiration is carried out using enzymes, and there's a whole series of enzymes because it is a small series of steps. And every cell needs to make its own ATP, so ATP is not transferred from cell to cell. And as ATP gets used, more ATP needs to be generated. So all cells need a continuous supply of ATP. And so what is this ATP required? Well, it's required for things like DNA and RNA synthesis. It's required in protein synthesis. It's required for things like active transport. Remember the sodium-potassium ion pumps. It's required in muscle contraction, as well as cell signaling and vesicle transport. So the use of ATP is circular in a manner, or cyclical as well. So that energy from metabolism, or that process of cellular respiration, will take ADP and that phosphate group and join them together to create ATP. That ATP gets used for cellular work, or provides the energy for cellular work, and that ATP will generate the ADP and phosphate. Basically, that third phosphate gets cleaved off of the ATP, turning it into ADP, and then you have that lone phosphate group. So then the process has to start again in order to create that ATP. So energy for all of these processes is supplied by ATP. Energy from ATP is going to be eventually converted into heat. And because heat cannot be reused for cell activities, it is lost to the environment. So as a result, cells require a continuous source of ATP in order to meet their energy demands. The main process in which ATP is generated is through what's called aerobic cellular respiration. This is a series of chemical reactions in which glucose is broken down in the presence of oxygen to produce ATP. In this process of aerobic cellular respiration, one glucose molecule will yield a maximum of 38 ATP molecules. So that's a lot of ATPs generated from one glucose. The diagram here shows just very generally the steps involved in glucose breakdown. So there is the first step, referred to as glycolysis, in which glucose is broken down into pyruvic acid. That pyruvic acid is converted into a molecule called acetyl coenzyme A, which then enters into the Krebs cycle, and that Krebs cycle generates these carrier molecules, electron carrier molecules, which are used in a process called the electron transport chain. And it's in this last step where the majority of ATPs get generated. Just note that the Krebs cycle and electron transport chain occur within the mitochondria, and glycolysis occurs in the cytoplasm. Glycolysis and the Krebs cycle generate a very small amount of ATP, and it is the electron transport chain that generates the majority of ATPs in cellular respiration. Now, cellular respiration can occur without the presence of oxygen, and this is referred to as anaerobic cellular respiration. So glucose can be broken down without using oxygen to yield a small quantity of ATP. So in situations where a cell may be oxygen deficient or deprived of oxygen, that cell can continue cellular respiration for a short period of time. This can also be useful when a rapid burst of ATP production is needed. So if a sudden supply of energy is needed, but not enough oxygen is there, a cell can use this alternative process to generate the necessary energy that it needs. It's also useful when oxygen supplies run out in respiring cells, or in environments that are deficient in oxygen. And the diagram here just shows where anaerobic respiration would occur. So in this first step of cellular respiration, where glucose is turned into this molecule called pyruvate, in the presence of oxygen, that pyruvate would enter into the mitochondria, where further steps would occur. So the Krebs cycle and electron transport chain would happen. In situations where no oxygen is present, that pyruvate can be used or put into an alternative pathway to now create other molecules that would help generate a small amount of ATP, or keep a small amount of ATP being generated. In animals, such as us, as that pyruvate gets metabolized, it generates a molecule or a compound called lactic acid. And lactic acid is what causes that muscle burn when you've been running too fast or too quickly, and is an indication that your cells have been respiring in the presence of or without any oxygen. And by generating that lactic acid, you're removing pyruvate from the cell, which can allow glucose to continue to be broken down to generate a small amount of ATP. In organisms like yeast, a different byproduct is generated in anaerobic cellular respiration. So in the case of yeast, they do not produce lactic acid, but they produce ethanol as well as CO2 as their byproducts. And that ethanol and CO2 has great use for us, or we take advantage of that process of anaerobic respiration. So, like I said, we make use of anaerobic respiration for a lot of food products. So a lot of cheese can be generated through anaerobic respiration. Sauerkraut is basically fermented cabbage. Things like beer is fermented barley. Soy sauce, is fermented soybeans, and these are all products that are generated through anaerobic respiration. So, for example, with beer, this is yeast placed in the presence of barley. The yeast start undergoing anaerobic respiration and start to generate ethanol, which is alcohol, plus carbon dioxide. Bacteria can also be used in this process of anaerobic respiration to generate food products as well. So the example that IB wants you to know where anaerobic respiration gets used is in that of baking. So the use of anaerobic cell respiration in yeast to produce ethanol and carbon dioxide in baking. So yeast is a fungus that can respire aerobically or anaerobically, and then in that process of baking, and where yeast gets used is in bread making. And bread is made by adding water to flour, kneading the mixture to make a dough, and then baking it. Now, yeast gets added in to create gas bubbles, giving the bread a lighter texture. After kneading, the dough is kept warm so that yeast can continue to respire. In this situation, the oxygen is quickly used up, and the yeast will start to respire anaerobically, generating carbon dioxide and ethanol. The carbon dioxide is what will cause the dough to rise and gives it that light texture to it. And the ethanol that is generated gets evaporated quickly during the baking process. So having a look at this, this is dough when it initially starts out, and then when you leave it for a while for the yeast to do their thing, to start respiring and respiring anaerobically, that dough will then start to rise and even double in size and will give you a very lighter textured bread. Now, in humans, as I said, we produce lactate instead of ethanol and CO2. So lactate production in humans, when anaerobic respiration, is basically used to maximize the power of muscle contractions. So when certain activities such as weightlifting and sprinting are being performed, they generally require anaerobic respiration. Aerobic respiration will generate the greater yield of ATP, but anaerobic respiration can supply ATP very rapidly. And that rapid ATP generation will enable us to maximize the power of muscle contractions. Now, anaerobic respiration in humans will also produce lactate. The lactate builds up quickly, and there's actually a limit to the concentration that the body can handle. This is that burn that you start to feel after you've been running, and then that burn becomes too much and you have to stop. So that's your body telling you there's too much lactate in your muscles and you need to stop running. The lactate must be broken down by oxygen, and this can take several minutes. So, it takes you several minutes to get running again, or get running, in any significant amount again. And this is giving your body time to break down that lactic acid that is built up in your muscles. One of the skills that IB wants you to be able to do is to be able to analyze the results from experiments involving measurements of respiration rates in germinating seeds or invertebrates using a device called a respirometer. Now, a respirometer is any device used for measuring respiration rates. There are many possible designs, but most will involve some kind of sealed glass or plastic container in which the organism or tissue is placed. So in the diagram here, there's a tube or container in which seeds have been placed. These are the seeds here that have been placed. And then there's some type of alkali compound that's present, such as potassium hydroxide, that needs to be there to be able to absorb the carbon dioxide that is produced. And this is usually in another tube separate from the seeds. And then there needs to be some type of capillary containing fluid connected to the container. So here we have a U-shaped capillary tube, and in the capillary tube is a fluid that will move or be displaced as CO2 gets generated from respiration. And as that CO2 is generated, it'll move that liquid in the capillary tube. Graduation markings placed by the capillary tube can give an indication of how much or what distance or what volume or what distance mostly that volume would have moved, and you can use that to measure the quantity of CO2 that has been generated. So a respirometer can be used to determine the respiration rate of different organisms. The effect of temperature on respiration could also be determined, and then respiration rates of active and inactive organisms can be compared as well. There's more to be read on page 127, so make sure you read that and make some notes about it. For the Nature of Science statement, which relates to this is now assessing the ethics of scientific research, so the use of invertebrates in respirometers in these respirometer experiments does have ethical implications. So when you're using any organism, there's always several questions to consider before doing an experiment. Ignore this should be on the previous slide. Can that organism be safely returned to their habitat? So when you're using an invertebrate in a respirometer experiment, when you're finished with that, using that invertebrate, can it be safely returned to their original habitat? Will the animal suffer pain or other harm during the experiments? And if so, what can be done to minimize that suffering or pain of that organism? And then can the risk of accidents to the animal during the experiment be minimized? So are there ways of ensuring that you don't accidentally injure the organism under study? And is the use of animals in the experiment necessary, or can the use of animals be avoided? Are there alternative means or alternative models that can be used to obtain similar results from the experiments? So that is the end of this video for subtopic 2.8 on cellular respiration.